Hello everyone, my name is Adam Bush, and it is an honor to present a synopsis of our work in cerebral oxygenation and sickle cell disease for this month's edition of MRM Highlights. So let's go ahead and begin. Here's an outline of the material that will be covered in this presentation. So for significance, our laboratory focuses on research in sickle cell disease, where sickle cell disease is the most commonly occurring genetic disease in the U.S., affecting approximately 100,000 Americans. Epidemiologically, having one copy of the sickle cell gene, or sickle cell trait, offers protection against the plasmodium carried by mosquitoes responsible for malaria. As a result, in the United States, the sickle cell gene is commonly found in people of African descent. Although sickle cell trait is benign, when a child inherits two copies of the sickle cell hemoglobin mutation, this gives rise to sickle cell disease, a lifelong, debilitating condition. Sickle cell disease is characterized by the production of an abnormal hemoglobin molecule, where hemoglobin is the oxygen-carrying protein found in red blood cells and is necessary to carry oxygen to tissues throughout the body. In sickle cell disease, the sickle hemoglobin will polymerize with other sickle hemoglobin molecules when deoxygenated, forming long, rigid strands that flex and misshapen the cell, putting stress on the cellular matrix. Under microscopy, polymerized cells can take the shape of a crescent moon or sickle, giving the disease its name. Sickle cells can become sticky and rigid, blocking blood flow, termed vasoocclusion. Frequent polymerization can also cause the cell to burst or hemolyze, releasing the cellular contents into the bloodstream, which are toxic outside the blood cell. Classically, the constant intracellular polymerization and hemolysis leads to low blood cell levels, called anemia, a common feature of the disease. Together, hemolysis and vasoocclusion results in clinical complications in essentially every organ system in the body. Perhaps the most devastating complications, however, are those that occur in the brain, in particular stroke. Stroke results from a lack of tissue blood supply or ischemia that leads to localized brain necrosis. Overt stroke, as seen in this T2 weighted image on the right, often causes significant morbidity, including paralysis or even death. Historically, about 11% of children with sickle cell disease will have suffered a stroke by the age of 20. As a result, this is the most commonly occurring pediatric stroke population. Furthermore, with the increasing usage of MRI, so-called silent strokes have been identified. These punctate lesions appear as hypointensities in T1 weighted images and hyperintensities in T2 weighted images, and are devoid of the classical neurological signs associated with stroke, and are thus considered, quote, silent, if you will. Roughly 40% of children with sickle cell disease will present silent infarctions by the age of 18, which has been associated with decrements in intelligence quotient, academic difficulties, and increased overt stroke risk. As I think is evident by these two volumetric images of two healthy 19-year-olds, silent stroke is hardly a silent disease. Thus, our group is interested in using MRI to study the mechanisms of cerebrovascular disease and sickle cell disease and develop tools that are better able to identify those patients at risk. And that all stroke is secondary to oxygen supply and demand mismatch, we chose to start by measuring cerebral oxygen supply and demand as a starting point for the generation of biomarkers of disease. Using a simple physiological relationship known as the Fick principle, we can define oxygen demand as a difference in oxygen inflow and oxygen outflow to the brain, where the product of cerebral blood flow and blood oxygen content, which is predominantly determined by the hemoglobin concentration and blood oxygen saturation, equates oxygen flow. Though many of these quantities are routinely measured clinically, historically, measuring venous blood oxygen saturation has been challenging and limited to invasive procedures like jugular catheterization. Now that we've covered the background and significance, let's move on to the methods used in this study. So the first method used in this study was phase contrast MRI for quantification of global cerebral blood flow. Phase contrast works by using a pair of bipolar gradients to encode the velocity into the phase of the image. Subtraction of two images with equal and opposite phase accrual is then used to isolate moving spins and calculate a velocity image. By performing 2D phase contrast in the neck, in calculating the velocity times the cross-sectional area of the 4 feeding vessels to the brain, global cerebral blood flow can be easily calculated. The next technique used in this study was T2 relaxation under spin tagging, TRUST. TRUST works by magnetically tagging blood in the brain. That blood is then allowed to flow into the sagittal sinus where an image is acquired. This was repeated without the magnetic tag, and the difference signal now is proportional only to the isolated blood, minimizing partial volume effects. T2 weighting is applied to the blood image using a preparation pulse train, and blood T2 is calculated. Predetermined empirically derived calibration equations relating blood T2, blood oxygen saturation, and hematocrit are then used to convert measured T2 values into oxygen saturation of the sagittal sinus. Though TRUST is a recently developed technique, this work is not the first to use TRUST in sickle cell disease patients, as there have been several publications. Fortunately, the findings from all these studies are largely the same, with each reporting that oxygen saturation values are lower and oxygen extraction and metabolism are elevated in subjects with sickle cell disease. The thought being that subjects with sickle cell disease are tapping into oxygen extraction reserves, possibly reflecting stroke risk similar to phenotypes observed in carotid stenosis. Unfortunately, all these works are limited in that they use an empirical calibration curve to convert blood T2 measurements into oxygen saturation values that is not applicable in patients with sickle cell disease for several reasons. For one, previous calibration curves were determined using cow or bovine blood, which has several distinct properties. For instance, bovine blood cells are half the size of human red blood cells and do not aggregate like typical human erythrocytes. Secondly, the original bovine models were defined over the hematocrit range normally found in healthy subjects, approximately 35 to 55%. Since sickle cell disease patients are anemic, they rarely fall into this range. A previous work from our group addressed both of these concerns by developing a calibration curve in hemoglobin A, human blood samples over a wide range of hematocrits. Here is the bovine model used by the previous papers, and here is our human hemoglobin A blood model. Though they demonstrate similar behavior over the healthy hematocrit range, when the bovine model is represented over a wider range of hematocrits, the nonlinear extrapolation errors become clear over an anemic hematocrit range. Although the hemoglobin A human blood model addresses both of these issues, it remains unknown whether the unique cell properties of the sickle cell disease, such as polymerization, hemolysis, and cell damage, also influence the calibration equations. So in order to address this question, we performed blood experiments on several subjects with sickle cell disease across a range of genotypes, transfusion statuses, hematocrits, and oxygen saturation levels. The experiments were tightly controlled for things like temperature, pH, and sedimentation to ensure that ex vivo conditions mimic the in vivo conditions as close as possible. Using the ex vivo system, we observed several interesting findings. On the left is the hemoglobin A model derived from our previous work, and on the right is our newly defined hemoglobin S model. The two key differences were a steeper dependence on oxygenation and a lack of hematocrit dependence in the hemoglobin S model. At this time, we are uncertain as to why the hemoglobin S model demonstrated higher relaxation with oxygenation, but possible explanations include increased permeability and/or polymerization. However, this is also the subject of future work. As for the lack of hematocrit dependence, this is likely the product of increased variability in the limited hematocrit range used in the study. Since sickle cell erythrocytes are quite fragile, we only measured red cells at their native hematocrits with the aim of minimizing hemolysis. However, another group has observed a loss of hematocrit dependence in blood T1 measurements in sickle cell disease patients, increasing confidence in our observations. To complete the study, we then made TRUST and phase contrast measurements in control subjects and subjects with sickle cell disease, allowing us to assess global cerebral oxygenation and metabolism. To the right is some of the demographic information of patients enrolled in this study. As expected, we found that if we use a bovine model, sickle cell disease subjects demonstrated similar venous saturation values compared to controls, which resulted in higher cerebral oxygen extraction values and cerebral oxygen metabolism. Surprisingly, however, if we use the newly derived Sickle model in our sickle cell disease subjects, the oxygenation was much higher than controls, which meant that oxygen extraction and metabolism were actually decreased in subjects with sickle cell disease, a complete opposite finding of previous work. Unfortunately, our experimental conditions were not identical to in vivo conditions, and factors such as increased hemolysis and/or polymerization ex vivo could be confounding our results. So, in order to build confidence in our methods and address the question of can we actually trust, we also performed an external validation. This included performing peripheral blood draws from the antecubital vein and comparing peripheral blood gas oximetry to cerebral truss oximetry. We found there was no relationship between the cerebral and peripheral oximetry when the bovine model was used. However, when the newly defined sickle model was used, there was a tight linear relationship. Furthermore, when the anemic subjects with normal hemoglobin A from our previous published work are included, this relationship is only strengthened, suggesting that both our anemic model and our sickle model are in fact correct in describing real physiology. But what does decreased oxygen extraction and metabolism mean physiologically in this population? At this time, we're not totally sure, but our working hypothesis is that of cerebrovascular shunting. So, in a healthy vascular bed, the bulk of gas and nutrient exchange takes place in the microvascular bed, where blood flow slows in the capillaries, allowing time for gas exchange to occur. On the other hand, in a diseased or high blood flow conditions, gas exchange does not happen uniformly. We hypothesize there are slow and fast conduit microvessels with variable oxygen concentrations. In slow channels, extraction is high and tissue is nearly ischemic. However, in high flow channels, extraction is low. Because the high flow vessels disproportionately contribute to the venous pool, global oxygen extraction paradoxically appears low. Meanwhile, tissues may still have local ischemia despite low blood extraction and a misleadingly large global oxygen extraction reserve. This phenomenon is similar to arterial venous anastomosis shunting, whether or not large conduit vessel shunts are actually present. Our future work aims to explore the validity of this shunting hypothesis and its physiologic, pathophysiologic, and clinical significance. So in closing, Truss MRI can measure cerebral oxygenation in sickle cell disease. Sickle cell blood exhibits a unique magnetic phenotype. Existing cerebral oxygenation litter using bovine models in sickle cell disease is incorrect. Global cerebral oxygen extraction and metabolism are diminished in sickle cell disease, and the shunting hypothesis is one possible explanation. With that, I would like to thank my previous colleagues at Children's Hospital Los Angeles, funding sources, the Hung Zan Liu Group, and my current group at Stanford University. Thank you.